Welcome, my name is Dr. Moreno, and I will be talking to you today about skeletal muscle, structure, and function. As you know, I am a mother of two, so if you hear noise in the background, those are my children. So let's dive in. This section on sport performance focuses on how the body moves effectively and intentionally during physical activity. These movements are made possible by the force produced by muscle, which acts on the skeleton through lower systems to move different parts of our body. The brain's cerebral cortex controls these muscles by sending signals through motor neurons with the help from the peripheral nervous system. For muscles to keep working effectively, they need a constant supply of oxygen, and nutrients and waste products like carbon dioxide must be removed. Tasks carried out by the cardiovascular and respiratory systems. To design effective training programs for our athletes and apply scientific principles to athletic performance, strength and conditioning professionals need a solid understanding of not just how muscles and bone work, but also how the body supports skeletal systems help maintain muscle function. So this chapter and the following chapters will provide an overview of the key roles played by the neuromuscular, musculoskeletal, cardiovascular, and respiratory systems in building and maintaining muscular strength and power. After reviewing this chapter, structure and function of body systems, you should be able to describe the differences among smooth, skeletal, and cardiac muscle, understand the basic structure of skeletal muscle, know the characteristics that differentiate fast twitch from slow twitch, and be familiar with the sliding filament model of muscle contractions. Here are also your keywords. If you have any questions on these, please feel free to reach out. Here's some additional review resources for you to utilize. I provided the musculoskeletal explained in 6 minutes. The authors of this video did an excellent job of providing an overview of kinesiology for you, and then also a few sliding filament theory illustrations demonstrating in 3D on how it all works together. Check those out. They may be useful as we go through and talk about the skeleton theory today. All about the musculoskeletal system. The musculoskeletal system of the human body consists of bones, joints, muscles, and tendons that configure to allow us to move on a daily basis. The muscles of the human body do not actually directly apply force to the ground, yet what they do is pull against bones and rotate about joints and transmit force to our environment. Our human body consists of more than 600 skeletal muscle. Compose that about 40 to 50% of our total body weight. Now think about that. 50% of our body weight is made up of muscle, which helps us move, generate force, pulls on the bone to allow us to perform those daily active living movements. Carrying our groceries from our car inside to the house, allowing us to grab the groceries from the bag and put them into the cabinets, the fridge, etc. Right? So therefore, we must be strong enough to complete those tasks. The skeletal muscle performs 3 vital functions. The first one, force generation for locomotion and breathing. Breathing is automatic. Think about that. We don't tell our body to breathe. It just naturally occurs. 2, force generation for postural support. We generate force, contraction, muscle actions, right? But we also have this tonic tone that is constantly on. Muscles in the trunk, right? Trunk skeletal muscles from our shoulders down to our hips or even our knees of who you may talk to. So check your posture. Check it right now as you're sitting and listening to this lecture. Make sure your shoulders are depressed down and back. Our ribs are stacked over your hips. That way we have less stress on the back. 3, heat production during periods of cold stress. We shiver By shivering, we produce heat. That's our skeletal system taking care of us. Skeletal muscle contracts rapidly to generate heat and elevate our core temperature. We have 3 different types of skeletal muscle, which we've discussed briefly, but we're going to dive in just a little bit more today. The first type, skeletal. This again is with the somatic nervous system, which means that it's voluntary, it's controlled consciously. We have to consciously send a signal from the central nervous system to the peripheral in order for this muscle to be contracted. And we have over 600 muscles throughout the body. Skeletal is attached to the skeleton and provides force for movement on the bony lever system. Remember, our skeletal system, the muscle belly, attaches to tendon, which does not produce a force. That tendon is then attached to bone, and the bone and the tendon go along for that passive ride when the muscle belly shortens on that muscle. It's innervated by a voluntary and that somatic nervous system, and again it consists of long cylinder muscle fibers. The cardiac is the heart, only in the heart. In all the vertebrae, it's a network of striated fibers. Now this is something that is very similar to the skeletal. Great test question. What is something that is consistent between the skeletal and the cardiac? They're both known as striated muscle fibers. And that's because of the muscle proteins that it contains. It contains 2 specific proteins that allow for contraction to take place for both skeletal and cardiac. These are known as myosin and actin. So again, the cardiac is involuntary, which is opposite. It does not require a conscious signal to be sent to the heart to beat. Therefore, it controls itself with assistance from the nervous system as well as the endocrine systems. The third type of muscle is known as smooth. Similar to cardiac, it is involuntary. It's controlled by local factors and the nervous system as well as the endocrine. Local factors meaning it's stimulated by the autonomic nervous system. So it's just, again, automatic. It'll take place as it needs to. It's a main job, among others, but primary job is to ensure that we have proper digestion and GI tract function. It does not, however, have a striated appearance like the cardiac and skeletal muscle. From here on out, we're going to be discussing skeletal muscle organization. On the handout that I'll be providing to you, you'll have a skeletal muscle organization. Although this is not the primary point that I want you to focus on, it's still a very important piece and a good refresher from kinesiology. We may not be diving in as deep as you did in kines, but again, a good reminder. So each muscle is surrounded by a connective tissue called the epinesium. The fascicle is a bundle of muscle fiber. This is surrounded by connective tissue called the perimesium. The endomesium is the connective tissue surrounding the individual muscle fibers, and they're packed within the fascicle. So that fascicle that's popped out is wrapped by the perimesium. This is our big old bundle of fibers packed together. The myofibrils are organelles within the muscle that contain thick and thin filaments. These are the myosin and the actin. Those myofilaments are those contractile proteins we were talking about. These are organized into what's known as a sarcomere within that individual muscle fiber itself. So let's back up just a little bit. That fascicle. Think of a bundle of straws. When you buy a bag of straws, you have the perimysium, which is the bag, and then you have this bundle as a whole, which is the fascicle, and then inside each individual fiber, or each individual straw, is known as a muscle fiber. We're going to talk about a single straw. We're going to cut it in half and open it up. That's what we're essentially doing for the remainder of this section. We're looking at how that individual cell, muscle cell, functions. Now, when we hear the word cell, we tend to think of a circle or a bubble-looking shape with organelles on the inside. We talked about cytoplasm. We discussed some DNA as well as a mitochondria. But for muscle cells, for skeletal muscle at least, look like an individual straw. We're pulling a single straw out, which is a myofibril, also known as a muscle cell. We're going to break it down, looking at it, to see what a sarcomere is, and those contractile proteins that live within that sarcomere. We just discussed the organization of a muscle. This is pretty much the exact same thing, but diving in a little bit deeper. Remember that fascicle is the bag of straws. We're going to pull a single straw out, which is that muscle fiber, which again is our cell. So let's look at the muscle fiber cell. You see in yellow the nerve. It's abbreviated as nerve axon branch. This is the nerve cell we talked about in the previous section. It's actually pointing to the axon itself. Of course, we can't see the myelinated sheath, the nodes around Vier, but we are now just bringing it back out so we can just see the bigger picture. The end of that neuron, known as that axon terminal or synaptic bulb, is now being abbreviated as motor endplate, which is the neuromuscular junction. Think back to the previous section. You know this definition. Write it out if you forgot it. Neuromuscular junction is simply the innervation between a nerve and a muscle. These do not touch. Remember, they don't physically touch. There's a synaptic cleft. There's a space or a gap in between the nerve and the muscle. Again, this communication is known as the motor end plate, neuromuscular junction. This tells the muscle to contract, how fast to contract, what specific type of the skeletal muscle needs to be recruited for that force production, which we'll talk about in a moment. There's a lot else going on in this photo, but I want to bring your attention to the individual straw, the myofibril. Again, the fascicle is the bundle of straws. And now we're going to be looking at that myofibril. The myofibril has what's known as sarcomeres. Towards the bottom right of your image, you have a sarcomere, and it's defined as a Z-line to Z-line. So in red, you can see these itty-bitty Z-shaped letters. Those are the Z-line to Z-line. We have a series of sarcomeres, one right after another, along this individual muscle fiber, the myofibril. Another definition for this sarcomere is the smallest contractile unit. So a sarcomere again is Z-line to Z-line, and the smallest contractile unit. Within each sarcomere, we have a dark and light filament. So it's illustrated here in blue and red. The blue, the dark, also the thick filament known as myosin. These are the myofilament contractile proteins that we were talking about earlier. Myosin and the thin filament, illustrated in red, known as the actin. Looking at the sarcomere image, the actin attaches to those Z lines. And the myosin is in the middle. The muscle fiber. Looking at an individual myofibril, we have a lot going on the inside. Again, we have a Z line to Z line, which is the sarcomere. Now we have another S word, which is different, known as the sarcolemma, that first bullet point. So please note, sarcolemma is different from sarcomere. Sarcomere is what we discussed on the previous slide, which is Z line to Z line. Sarcolemma is what's known as the plasma membrane that wraps around the muscle fiber itself. So on this outer layer that wraps around all of these myofibrils, that's known as the sarcolemma. Its purpose is to allow the action potential to travel along the sarcolemma quickly to be able to get inside the muscle to communicate contraction. The muscle fiber has multiple other systems that allow the action potential to travel throughout the muscle. The sarcolemma, as we just mentioned, allows the action potential to travel in opposite directions along the muscle. But how does that action potential get inside that myofibril? It gets inside through a single opening called the T-tubules. Transverse tubules, but we can call the T-tubules for short. It's illustrated here in gold. You see a little hole opening at the surface of the sarcolemma and the T-tubule, which almost makes like a T letter. So the T-tubules runs vertically straight down, all the way down, which is directly connected to this rest fibering on either side of the T-tubule, which is known as the sarcoplasmic reticulum, abbreviated as SR. So the T-tubules are connected to the sarcoplasmic reticulum. Lastly, on this slide, we have what's known as the mitochondria, which we know as the powerhouse of ATP, illustrated here in green. And of course, nucleus, because every cell must have a nucleus to hold that DNA. Some key points. There are 10,000 to more than 1 million fibers per muscle. We're again talking about a single muscle fiber. So muscle fiber unequivocally means muscle cell. So if we use those interchangeably, please don't get confused. Muscle fiber is a muscle cell. A muscle fiber is enclosed with a plasma membrane called the sarcolemma. Remember, this is different from the sarcomere. T-tubules form a network around fibers. They are the extension of the sarcolemma. This is because the T-tubule connects directly to the sarcolemma. It is the opening that allows the action potential to travel from the outside of the muscle into the muscle for force production to take place.
It allows for the transport of substances throughout the muscle, as serves as as well as serving to transport nerve impulses. Attached to the T-tubules, we have what's known as the sarcoplasmic reticulum, SR. This webbing stores calcium.
Throughout the years, different researchers have devised different schemes to classify skeletal muscle fibers. In general, two distinct fiber types have emerged for classification, termed fast twitch and slow twitch, type 2 and type 1, respectively. A fast twitch motor unit is one that develops force and also relaxes rapidly, and therefore has a short twitch time. In comparison, slow twitch motor units develop force and relax slowly. Therefore, they have a long twitch time. So histochemical staining for myosin ATPase content is often used to classify fibers as slow twitch or fast twitch. Although the techniques can stain for multiple fiber types, the commonly identification are going to be our type 1, again, that's slow twitch, and type 2 are fast twitch. As you can tell in this image here, this is how we're differentiating the two types of fibers. You can see that type 1 is our smallest in diameter. Again, these are slow to contract and slow to relax. And then as we go along this continuum, even though we have these specific types of type 1, type 2, remember, muscle is on a continuum. Diameter of muscle gets larger over time. So type 2A gets our intermediate, and then type 2X is our largest, our quickest fiber type. Proportions of each type of muscle fibers are going to vary from person to person. Also, muscle to muscle. Certain muscles, such as the gastrocnemius, will have a higher content of type 1 fibers. Our trunk musculature will have a higher content of type 1 fibers. However, for instance, for some individuals, our quadriceps may have a greater amount of type 2 fibers. It's going to vary from person to person. If you take specific athletes, marathon runners versus our sprinters, their body compositions are drastically different because of the performance that they need to accomplish. So only one type of slow twitch, type 1 muscle fiber exists in humans. So let's talk about type 1 fiber a little bit more. So type 1 fibers are generally efficient and fatigue resistant and have a high capacity for aerobic energy supply. And they have a limited potential for rapid force development as characterized by low myosin ATPase activity and low anaerobic power. When we compare this to type 2 motor units, they're essentially the opposite, characterized as inefficient and fatigable, and having a low aerobic power, rapid force development, high myosin ATPase activity, and high anaerobic power. So type A and X, so type 2A and type 2X fibers, differ mainly in the capacity for aerobic and oxidative energy supply. So let's dive in a little bit more on that. So type 2A fibers, for example, have greater capacity for aerobic metabolism and more capillaries surrounding them than the type 2X. So therefore, they're showing a greater resistance to fatigue. So remember, along this continuum, type 1, we're starting at very low fatigue. As we go down this continuum, that fatigue rate gets faster. We can't hold off on fatigue as long. So again, based on these differences, it is not surprising that the postural muscles, such as the soleus, the gastroc, have a higher composition of our type 1 fibers, whereas large, so-called our locomotive muscles, such as the quadricep, as we mentioned earlier, have a mixture of type 1 and type 2 fibers to enable both low and high power output activities, such as our jogging, sprinting. Provided an additional example, a 100-meter sprint. Think Usain Bolt. He has a lower amount of type 1 fibers to complete this task, whereas he might have a incredibly high type 2 amount of fiber. Whereas we have a marathon runner who's going to have a high relative involvement of muscle fiber types from the type 1 and low for type 2. Tennis may have a high involvement for type 1 and type 2. Think of the performance, think of their activities. We need to create an analysis of their sport. There are times where they're going to need a high power output and times where they'll need low power output. As we were just talking about the differences between the type 1 and type 2 fibers being able to produce low and high power outputs based on their activity, well, here's a table illustrating contraction time, oxidative capacity, so how much oxygen that is delivered to the skeletal muscle, diameter, how large, the resistance to fatigue, and generate force. So this is a summary again of primary characteristics of fiber types. So motor units are composed of muscle fibers with specific physiological characteristics that determine their functional capacity. So again, type 1 is slow twitch, type 2 is fast twitch. So throughout the day, we can determine that muscle can give us force output according to the level required for that particular task. For instance, carrying a single bag of groceries in versus all of them at once plus carrying children. The latter will require more force output. Under the bar with 100% of our 1 RM, we're going to require a great deal of force output. Body weight squats may not require a lot of force output. This ability to vary force is essential for performance to ensure that we have smooth, coordinated patterns of motion. Muscular force can be graded in 2 ways. So one, through a variation of frequencies at which motor units are activated. This is known as our firing rate. So if a motor unit is activated once, the twitch arises and does not produce a great deal of force. However, if that frequency of activation is increased so that the force of that single twitch begins to overlap, so now we have multiple twitches occurring, the resulting force evolved. Therefore, the motor unit is a greater method of bearing a larger force output. Especially important for smaller muscles, such as in the hand. Even at low forces, most of the motor units in these muscles are activated, although again at low frequencies, not able to produce a great deal of force, such as a 1 RM. Force output, the whole muscle intensifies throughout the increase in the firing rate of an individual motor unit. So in other means, the varying of skeletal muscle force involves increasing the force by varying the number of motor units activated, a process known in large muscles such as the quad. So looking at the diagram, if we start out walking and then begin to start jogging, additional force is needed. As in sprinting, at the end of a race, the fast twitch motor units are called into play to increase the pace. Unfortunately, exercise at such an intensity can't be maintained for a very long period of time. Therefore, our type 2X fibers are fatigable. Therefore, you see under type 2X and resistance to fatigue, it's low. Another example requires near maximum performance would be a power clean. Most of the motor units are called into play with fast twitch units making more significant contributions to force output. One important item to remember, that complete activation of available motor neurons is probably not a possible thing to achieve in untrained individuals. Their neurons are not trained, they're not capable of recruiting that high order of fibers. So, in summary, the force output of a muscle can be varied through change in the frequency of activation of individual motor units or change in the number of activated motor units recruited. An endurance athlete with predominantly type 1 in their leg will likely have a high percentage in the arms as well, similar for those with a high amount of type 2 fibers. So type 1 and type 2 are not the same in all muscles of the body. We mentioned this. Most muscles in the body contain a mixture of 50% fast and slow twitch. The general population will probably have 50% of both fiber types. This is going to be influenced by genetics, blood levels of hormones, individual exercise habits, and training status. Unfortunately, as much as we would love to pick our own sport, our sport chooses us. As a short individual, I am more prone to weightlifting, gymnastics, whereas if I wanted to play basketball, I could absolutely play. However, the abilities that someone has as a taller individual may outplay me on the court. So again, genetics are going to heavily influence the amount of type of fibers that we have. So fiber type plays an important role in performance in both power and endurance events, right? For instance, if we are looking to run a marathon, type 1 fibers are going to be primarily the recruited muscle, type 1 fibers. Whereas if we are at a power event, such as a weightlifting, where we're doing the clean and jerk or snatch, type 2 fibers are going to be highly recruited, as we are going to need maximal strength and power output to complete these tasks. Again, we mentioned earlier that there are muscles that are composed of very high percentage of type 1 for everyone. This, again, is an example of the soleus muscle, where it's tonically on. We need this for posture to remain upright. Same in our trunk. Our trunk musculature is also going to help us maintain that upright position, making sure that our ribs are stacked over our hips to maintain an upright position throughout the day. Previously, we mentioned that motor units are composed of muscle fibers with physiological characteristics that determine their functional capacity. So force production differences are different between type 1 and type 2 fibers. Less force output occurs with type 1 fiber. However, motor units made up of motor neurons or nerves and all the fibers that it innervates. So think of having a bundle of straws together, wrapped into a string, right? A single fiber is a single straw. A single straw cannot fire individually. The entire bundle must fire together. Motor unit is composed of a single alpha motor neuron and all the muscle fibers that it innervates. So an alpha motor neuron determines whether a fiber is type 1 or 2. So a cell body is smaller for our type 1 versus our type 2, right? So we have this collection of signals in our cell body, and we'll break down a neuron in a little bit. But type 1 innervates less than 300 muscle fibers, whereas type 2 innervates greater than 300 muscle fibers. Therefore, we're able to produce a great deal of more force with larger straw bundles, if you will, motor units. So we know skeletal muscle is an organ that contains muscle tissue, connective tissue, nerve, and blood vessels. So let's talk about the nerve. So this is a structure of the motor neuron, right? So we have the cell body, the dendrites, which are the spider-looking arms that receive the messages from the central nervous system to the peripheral nervous system and transfer them down to the cell body. From the cell body, that message is then sent along that axon. And we have this myelin that's in blue, that covers our axon. And the space between the myelin sheath is what's called the node ramifier. So the thicker this myelin, the faster this message can be delivered down to the axon terminal and the synaptic knob to then be delivered to the muscle, which we'll talk about in a few slides. So again, the central nervous system sends a signal to the peripheral system, which is received by the dendrites. And once we get enough of those signals, enough of that message can then be delivered along the axon, through the axon terminal to the synaptic knob, and then eventually to the muscle. The interior structure of a muscle fiber has a sarcoplasm, which is the cytoplasm of a muscle fiber, containing contractile components consisting of protein filaments and other proteins, stored glycogen and fat particles, enzymes, and specialized organelles such as the mitochondria and sarcoplasmic reticulum. Hundreds of myofibrils dominate the sarcoplasm. Myofibrils contain the apparatus that contracts the muscle cell, which consists primarily of two types of myofilament, myosin and actin. The myosin filament consists of a globular head, a hinge point, and a fibrous tail. The globular head protrudes away from the myosin filament at a regular interval, and a pair of myosin filaments forms a cross bridge, which interacts with actin. The actin filament consists of two strands arranged in a double helix. The myosin and actin filaments are organized longitudinally in the smallest contractile unit of a skeletal muscle, known as a sarcomere. A sarcomere averages roughly 2.5 micronmeters in length in a relaxed fiber. So again, the structure of a myofibril has multiple sarcomeres longitudinally along a fiber, which contains a thin filament, which is our actin, our thick filament, which is our myosin, and when these two interact, we are able to then produce force. The myofibril key points. So the sarcomere, again, are multiple sarcomeres along this fiber in a longitudinal, horizontal fashion. It's composed, again, of our thick and our thin filaments, which are the myosin and actin, respectively. They're responsible for muscle contraction and the striated appearance of the muscle. Smallest contractile unit of a muscle is a sarcomere. So a myosin head, again, it has a globular head at one end. Think of your forearm and your fist. If you were your hand and it made a fist, your fist is the globular head, and then you have a tail for its myosin. This is going to attach. The globular head will attach to the actin, pull on the actin, causing the sarcomere to shorten like an accordion. This, in theory, is our force production. This image is a detailed view of our sarcomere. Remember, our sarcomere is the smallest contractile unit. These run from Z-line to Z-line, illustrated in the red on the outside of our image on our fiber. So again, we are looking at a single sarcomere, and these run the entire length of our fiber. So when we talk about force production, what's actually happening within the muscle? Think of an accordion. When force production occurs, those Z lines move towards that M line right in the middle. Think of an accordion going in and out. What causes that Z line to move in and out? Let's look at the myosin. So the myosin in red has a globular head. Remember that fist and forearm example I provided. Your fist, which is the globular head, attaches to the actin. So the globular head moves and attaches to the actin and then pulls towards the M line. If we looked at how the actin is laid out, it attaches to that Z line. So when the globular head of the myosin attaches to actin, it's now connected and it's going to pull the actin towards the M line, in turn also causing the M lines to move inward like an accordion. As an accordion makes noise, the muscle is going to create or produce force. So when our gobbler head connects to the actin, it is known as a cross bridge. This connection is a cross bridge. The number of cross bridges that are formed between actin and myosin at any instant in time dictates the force production of a muscle. As previously mentioned, when our myosin head pulls on that actin, this is known as the energy for pulling action, or known as our power stroke. In order for our myosin head to be able to attach to our actin, a binding site must be revealed. In order for a binding site to be revealed, we have calcium binding sites on the troponin. Calcium binds troponin, causing the tropomyosin, the string, the pink-like string, to shift, revealing binding sites for the myosin head to connect to then create this cross bridge, then pull the actin into a power stroke, creating force. So as a reminder, the motor unit comprised of a motor neuron and all the fibers it connects to. The motor end plate is the junction where the sarcolemma forms a pocket for muscle fiber innervation from the alpha motor neuron, also known as our neuromuscular junction. So a neuromuscular junction is when the nerve connects to our muscle. So let's talk more about this action potential from the neuron to the muscle surface. So again, muscle fibers are innervated by the motor neurons that transmit these impulses in the form of electrochemical signals from the spinal cord to the muscle, from the central nervous system to the peripheral. A motor neuron generally has numerous terminal branches at the end of its axon, thus innervating many different muscle fibers. Again, our neuromuscular junction, as illustrated here in this image. We have our neuron that is going to be communicating with the muscle, neuromuscular junction. So when a motor neuron fires an impulse, or our action potential, all of the fibers that it serves are simultaneously activated and develops force, which is that cross bridge to our power stroke interaction. But in order to get to the power stroke, we have to talk about these steps that allow us that power stroke and force production to take place. So muscles that must function with great precision, such as eye muscle, may have motor units with as few as one muscle fiber per motor unit. Changes in muscle in the number of active motor units in these smaller muscles can produce the extremely fine gradients in force that are necessary for precise movements of the eyeball. However, for larger muscle groups, such as the quads, glutes, require less precision and may serve hundreds of fibers served by one motor neuron. So this action potential that flows along the motor neuron is not capable of directly exciting the muscle fibers. Remember, we said that we had a power stroke that creates this force production that connects the myosin to the actin. So instead, the motor neuron excites the muscle fiber that innervates the chemical transmission. So the arrival of the action potential at the nerve terminal causes a release of this neurotransmitter, acetylcholine. So number one, our action potential travels down the axon. It moves into the end bulb, which then causes this release of calcium to enter the bulb, signaling our acetylcholine, ACH, to be released from the storage vessels. You see this ACH going into the space between the neuron and the muscle, which is on the motor end plate. So ACH is then released into this neuromuscular junction, this space, travels across the synaptic cleft, and the calcium binds to these ACH receptors on that motor end plate, our muscle. So 2 ACH molecules are needed to activate the receptors. This then causes depolarization of the sarcolemma, an electrical current, the action potential moves in both directions along the muscle to eventually get to the cross-bridge interactions. So, fun fact or question, why take smelling salts for weightlifting? Why do we use these? So the answer is to increase the number of active motor units by increasing the number of graded potentials, causing hypopolarization, membrane potentials. As mentioned on the previous slide, once a sufficient amount of acetylcholine is released, an action potential is generated along the sarcolemma and the fiber contracts. So all of the muscle fibers in the motor unit contract and develop force at the same time. There is no evidence that motor neuron stimulus causes only some of the fibers to contract. This is also known as the all or none principle of the muscle. So as that action potential travels along the sarcolemma and moves into the interior of the fiber by traveling through the transverse tubules, illustrated here in purple. So the action potential travels along that purple illustrated line down into the muscle through the T-tubules and signal the sarcoplasmic reticulum, abbreviated as SR, to be opened. The sarcoplasmic reticulum, the SR, once opened or signaled to be opened by this electrical signal, causes calcium to be released. So the calcium voltage gates are opened as a result of the action potential traveling into the muscle. Those voltage gates open, calcium is released, and then continues along this process. Once calcium is released from the sarcoplasmic reticulum, it binds to the thin protein. Remember, the thin protein is built of actin, troponin, and tropomyosin. Now, in order for a cross bridge to occur, the actin, the troponin, and tropomyosin, the thin filament, needs to shift. So again, calcium is released from the sarcoplasmic reticulum. That calcium is going to bind to troponin. That will then signal the tropomyosin to shift along the actin, revealing the binding sites on actin, then allows for myosin to attach the gobbler head to those binding sites to cause a cross-bridge interaction. So let's review. We're talking about the discharge of an action potential from the motor neuron, signal the release of acetylcholine from the motor end plate, crossing the neuromuscular junction, binding onto the motor end plate on the muscle. Once we have acetylcholine bind to the motor end plate, it causes a depolarization of the muscle. The action potential goes along the muscle, then into the T-tubules to enter interiorly of the muscle, causing a voltage change to the sarcoplasmic reticulum, allowing calcium to be released. Calcium then binds to troponin, causing a shift of the tropomyosin, which then reveals the binding site for the myosin to bind to actin, which is a cross bridge, then allows that myosin head to pull the actin towards the M line, ultimately pulling those Z lines closer together to produce force. This image is demonstrating a contraction of a myofibril in a stretched muscle where the I band and H zones are elongated, and there is a low force potential due to reduced cross bridge and actin alignment. Here, when muscle contracts, partially the I band and H zones are shortened, force potential is high due to the optimal cross bridge actin alignment. And fully contracted muscle force production is low because the overlap of actin reduces the potential for cross bridge actin alignment. So think of a bicep curl, for example. When you have your elbow in an extended position, it's relaxed. If you flex your elbow and your bicep concentrically contracts into a 90-degree position, you're contracting. Now, if you bring your wrist all the way up to your shoulder, you are fully contracted. So therefore, your bicep can no longer produce any additional cross-bridge interaction to produce greater force at this point. So again, in simplest form, the sliding filament theory states the actin filament at each end of the sarcomere slide inwards on the myosin filament, pulling the Z lines towards the center of the sarcomere and therefore shortening the muscle fiber. So as actin filaments slide over the myosin filament, causing the H zone and I band to shrink, this action of the myosin cross bridge pulling on the actin filament is responsible for the movement of actin. And because only a very small displacement of actin filament occurs with each flexion of the myosin cross bridge, a very rapid, repeated flexions must occur in many cross bridges throughout the entire muscle for measurable force and movement to take place. So while we are talking about a singular myosin gobbler head attaching to a binding site, think of multiple arms simultaneously going right after one another in order to continue force output. And again, this will continue for an extended period of time. So in review, the myosin cross bridges are activated once the myosin gobbler head attaches to the binding site along the actin. The change in the cross bridge causes the myosin head to tilt towards the arm of the cross bridge and drag the actin and myosin filaments in the opposite directions. The tilt of the myosin head is known as that power stroke. The pulling of the actin filament past the myosin results in the muscle shortening and generation of muscle force. So in order for the sliding filament theory to take place, in order for force production to occur, three things are absolutely required. One, action potential, that electrical stimulus that goes down the neuron, through the motor end plate, across the sarcolemma, down the T-tubules, that releases calcium. Number two, calcium is also required. Calcium then is released from the sarcoplasmic reticulum, binds to troponin, causing the shift to change of the thin filament, revealing the binding sites on actin. And the last thing that is required for force production, energy, ATP. So how do we generate energy once the nutrients are broken down, right? This is known as our ATP-ADP cycle. So this cycle provides the storage and use of energy in living things. Energy is defined as the ability to do work. During the contraction phase, the energy for pulling the power stroke occurs from hydrolysis, the breakdown of ATP to ADP, a reaction catalyzed by the enzyme myosin adenosine triphosphate, ATPase. Another molecule of ATP must replace the ADP on the myosin cross-bridge gobbler head in order for the head to detach from an active actin site and return to its original position. So let's talk ATP, adenosine triphosphate. This is the immediate source of energy for muscular contraction, high energy phosphate compound, ATP. So energy transfer in the body occurs via the release of energy trapped within chemical bonds of various molecules. So formed by combining adenosine diphosphate, ADP, and an organic phosphate. So bond held together by a very large amount of energy. So there are three main parts of this. The adenosine portion, the amino acid, ribose, which is a 5-carbon sugar, and 3 phosphate groups. In between the phosphate groups is the energy once it's cracked. So think of a pencil breaking. Once you hear that pop from the pencil breaking, that is a release of energy. The same thing with the 3 linked phosphate groups. Once each of those bonds are broken, it releases energy. So let's talk about the breakdown of ATP. ATP, right? So we have 3 phosphate molecules broken down into ADP, which is diphosphate, for 2 phosphate molecules. The release or break of one of those phosphates releases energy. So energy is available for cellular work and chemical synthesis. So in order for this to take place, the ATP splits by an enzyme called the myosin ATPase. So each molecule of ATP provides approximately 7 to 10 calories of energy for muscular contraction. And then it rebuilds back into our ATP. So measurable muscle shortening transpires only when the sequence of events, binding of calcium to troponin, coupling of the myosin cross-bridge with actin, power stroke, and then the dissociation of actin and myosin, that resetting the myosin head position is repeated over and over again throughout the muscle fiber. This occurs as long as 3 things are available. Calcium is available in the myofibril as due to the release from the sarcoplasmic reticulum. ATP is available to assist in uncoupling the myosin from the actin, and sufficient active myosin ATPase is available for catalyzing the breakdown of ATP. So let's talk about the process of actin and myosin binding an enzyme called myosin ATPase. So the function of myosin ATPase is to break down ATP. Now, a reminder that ATP binds to the myosin cross-bridge. So ATPase is an enzyme that splits ATP to release energy that will drive this power stroke, drive the contraction. So there are a couple of muscle type differences. Type 1 has a slower form of myosin ATPase, where type 2 has a fast form of ATPase. So therefore, ATP splits more rapidly for type 2 fibers. In turn, cross-bridge cycling also occurs at a much faster rate. As you can imagine, type 1 and type 2 have differences in their speed of contractions due to the form of the myosin ATPase. So ATPase breaks down ATP. The speed of the breakdown determines muscle speed. Also determines the rate of energy used. So the faster we move, the more energy we're going to utilize. Movement requires a lot of ATP, All right, so let's break this down. Number one, myosin head attaches to the actin myofilament, forming that cross-bridge interaction. So the myosin head is a high energy configuration. We have the ADP and PI because we exposed one of the phosphate molecules in order to attach. Now in step 2, we're going to use the ADP, and it's going to be able to allow us to generate that power stroke that allows the actin to pull on the Z line to move towards the M line. We're going to then need a new ATP to attach to that cross-bridge interaction to allow the gobbler head to detach from the actin. Then we're left with ADP in step 4. That ATP splits into the ADP. The myosin head is energized, and then it recocks back to its original position. So again, step 1, an ATP molecule is broken, and the energy is used to allow the myosin head to attach to the actin's binding site. The remainder of the ADP and PI will be used during the power stroke during step 2. Step 3, a new ATP is connected to that gobbler head and allows the detachment of the myosin head from the binding site. The ATP is split by the ATPase into ADP and PI. Therefore, the myosin head is energized and then can recock back into its original position, going back to step 1 to be able to reattach, power stroke, detach, recock back to its original position. And this, again, will continue as long as we have our calcium and enough ATP and enough of the action potential that's continuously being sent from the central nervous system down the motor neuron, through the motor end plate, through the sarcolemma, down to the T-tubules that releases the calcium binding to troponin signaling the tropomyosin to shift along the actin, revealing the myosin binding sites, then allowing for energy to be a part of the whole system, allowing for cross-bridge interactions, power stroke to generate force. So as we were talking about the cross-bridge cycling steps, we're just placing this into words now, where the myosin attaches to the actin once the binding sites are revealed on the thin filament. So once myosin attaches to actin, this is again called cross-bridge. Once we have a cross-bridge, we initiate a power stroke, the force production. This is due to when we release a phosphate generated in the previous contraction. The myosin head pivots, and the ADP is then released. From there, we need a new ATP to bind to the myosin. The myosin then detach from actin. The myosin head moves farther down the actin to reattach. But in order for that to take place, we need, again, ATP hydrolysis to occur once more. So we break the ATP into ADP due to the ATPase, releasing the inorganic phosphate generated in the previous contraction. Therefore, the myosin head pivots back into its original cocked position, allowing for additional cross-bridge and power strokes to take place. So we have talked about what happens during contraction and recharge when we detach, recock, and restart the whole process all over again. But what happens after? During the relaxation phase, relaxation occurs when the stimulus of the motor neuron stops. So this is our action potential. When there's no more or not enough of that action potential, that electrical stimulus, calcium is then pumped back into the sarcoplasmic reticulum, which then requires the tropomyosin to cover the binding sites on the actin, preventing the link between actin and myosin filaments. So therefore, the cross-bridge interactions can no longer take place. So relaxation is brought about by the return of actin and myosin filaments to their unbonded states. So when action potential is no longer present in the muscle fiber, relaxation occurs. Calcium detaches from troponin and goes back to the sarcoplasmic reticulum. The tropomyosin returns to its normal shape and covers the binding sites on the actin. This then prohibits any further cross-bridge cycling to occur, so no more force can be produced. So what are the three things that are required for force production? 1, action potential, the electrical stimulus. 2, calcium from the sarcoplasmic reticulum. And 3, energy, ATP.